Traditional Chinese medicine, TCM, is part of Hong Kong's culture. We can easily spot an herbal tea shop while hanging around. However, traditional Chinese medicine is more than just herbal tea. It includes acupuncture, tai chi, and herbal medicine. Herbal medicine is now a new direction in drug discovery. In fact, scientists suggest that it can be used to treat cancer, stroke, and cardiovascular diseases. One famous example is ginseng. It reduces inflammation, improves brain function, promotes the immune system, and reduces fatigue. As you all may know, herbal medicine needs to be brewed. In a modern society like Hong Kong, spending hours on brewing herbal tea is too time consuming, which doesn't fit our need. Herbal medicine is also bitter and contains a strong odor, which is why children often refuse to drink it. We need something convenient, something that allows people to skip the long brewing process and get rid of the bitterness. What can it be? As we concern about our health more, the demand for health supplement rises. Everyone wants to be healthy. Some turn to TCM, leading to an average industry growth of 10.6% in 5 years' time. Its total revenue in 2019 is $44.5 billion. Others turn to dietary supplements, which results in a compound growth rate of 8.2%, with a market size of $123.3 billion in 2019. How about combining the two, converting TCM into pills of health food supplements? With the advance in technology, we now have high contact screening technology to screen for active ingredients. It can be used to identify interactions between TCM and targeting cells, pinpoint biological changes in cells, predict toxicity of product, and study the related metabolic pathways. It is a combination of automated fluorescence microscopy and automated image analysis. Basically, we first generate an image and we take the image for analysis. First, we have an indicator which can indicate interactions between TCM and target. The indicator absorbs a photon and emits one later, but that one contains less energy than the first one. The less the energy is, the longer the wavelength is. If you look at a graph, red-colored light has the longest wavelength. Therefore, if we see areas red in color in the image, that indicates interactions between TCM and target cells. After identifying how the active ingredients interact with cells, of course, we have to extract them. We have the brewed herbal tea as shown in the graph on the right. Next, we add solvent to it. Usually, we use alcohol as solvent. The solute now dissolves in the solvent, and there are two layers of liquid. Collect the solid layer, and now we have our active ingredients. Although we already have the ingredients in hand, we still need to separate them. Chromatography is a good option. It can separate compounds based on their sizes, absorbance, charges, etc. A very common practice is to use column chromatography. In the column shown in the picture, there is a stationary phase. When mixture of compounds passes through the stationary phase, each of them adhere to it for a different time. Different compounds are eluted out and collected separately. The separated compound will be examined with mass spectrometry to deduce their formula. Take a look at the figure. The peak on the right indicates the mass to charge ratio of the compound, whereas the other peaks indicate that of its ions. We can either synthesize our product through biotransformation or flow chemistry. In biotransformation, the ingredients from the previous separation are incubated with enzymes, microbes, or fungi. They all convert compounds in ingredients into active ingredients. This method is more environmental friendly. If we choose to produce active ingredients chemically, we need to know their synthesis pathways. We'll just leave that for the computer to predict. With flow chemistry, mass production of active ingredient is a piece of cake. Reagents are pumped through tubes with a known rate and are rapidly mixed. This creates a high surface area to volume ratio, which greatly increases the yield. Artemisinin is one of the products made by flow chemistry. Before bringing the product to the market, clinical trial is necessary to study the actual effect on human beings. Though the product is tested on animal models, we need to be sure that the product is not harmful. Unlike trials done on drugs, health volunteers are recruited instead. The healthy volunteers will be randomly divided into two groups, one taking the actual product, one taking placebo. None of them knows what they are taking. This eliminates placebo effect. After the product is on the market, we still need to maintain its quality. We have to make sure that the active ingredients are the same as the original TCM. We need to control the concentration of the active ingredients at a safe level. The product mustn't be contaminated. The pill should be of the same size, color, odor, etc. Chromatography and mass spectrometry can be used to identify the active ingredients. Here's the reference used in my research. Research on development of health food supplement is still on the go. Once it succeeds, it will be a great game changer that allows mass production with lower cost and high efficiency, supplying a large quantity of cheap health food supplement to the market.